Assalamualaikum viewers. I hope you all are doing well, and I am Bushra Umar, the student of biotechnology from Jinnah University for Women, and I am the presenter at Sin Biotechnologies Association. And today I am going to give you a presentation about the topic biosensors in environmental monitoring. I would like to pay a heartiest thanks to Sin Biotechnologies Association for providing me the platform to express my scientific knowledge and to discover the areas of the science that are still unknown to me and to expand my knowledge of scientific world. So before jumping to the main topic, I want to introduce you to the word biosensors. The word biosensors is made up of two other words, bio and sensors. Bio means life, they are related to some living organism, and sensors mean something which can sense or detect. So what are biosensors in medical terms? As I mentioned, biosensors is defined by IUPAC, which is International Union of Pure and Applied Chemistry. And according to IUPAC, biosensors is a device that consists of living organism like an enzyme or can be a microorganism which can detect or sense any analyte or particle. And the biosensor is made up of biological recognition element, which is biological receptor, and it also consists of a transduction element which plays a crucial role in recognizing the analyte signals. And I discussed earlier that biosensors consist of living organisms like enzymes, tissues, and microorganisms. Environmental biosensors are slightly different from the traditional biosensors because we link these sensors like environmental biosensors with the detection of environment related particles or analytes. So in this slide, I am going to discuss 2 points. One is the example of biosensors that is commonly used for toxins detections, and the second one is the use of technologies that are applied for the making of biosensors, particularly environmental biosensors. So, as I mentioned, choline esterase enzymes example. So, basically, what are choline enzymes? These are the enzymes which catalyze the hydrolysis of neurotransmitter acetylcholine. And why we are using these enzymes? Because of their rapid ability to hydrolyze the compounds. Which type of compounds does it hydrolyze when they are using the biosensors? Well, they use they are used for the fast detection of cholinergic compounds. What are cholinergic compounds? These are the toxic compounds such as organophosphates, and they are found in the pesticides. Now moving towards the technologies that are used in making the biosensors. So as I mentioned here, silicon and thick film technologies are the strongest candidates for low cost disposable biosensors. Why we are using these two technologies? The thick film technology can operate in extreme heat and underwater. That's why we are using this technology. And silicon is the most common technology that is applied to the biosensors for the detection of analyte. Thick film means they consist of 6 to 8 successive layers of both conductive and resistive layers onto electrically insulating substrate, which means they have the ability to become positive and negative electrode which help in the detection of analyte. And the optical technologies are also used which are based on the principle of fluorescence, and the optical biosensors work on the principle of surface plasmon resonance, as I mentioned them as SPR, which is a phenomena that occurs when electron in a thin metal sheet become excited by light, and this light help to indicate the presence of a particular analyte. And the optical biosensors also work on the principle of interferometry, which is the measurement that includes certain characteristics of the waves themselves and the materials the wave interact with. So now we are going to learn about the types of biosensors, which are classified on two bases. One is transduction principle, such as optical or electrochemical, and the second classification is on the basis of recognition element, like the organism or element we are going to recognize. So on the basis of transducing element, the types of biosensors are optical biosensors, electrochemical biosensors, and mass-based biosensors, which are further classified into magnetoelectric and piezoelectric biosensors. Starting from the optical biosensors, these are the most common and basic type of biosensor that are being used. And what kind of analyte they consist? They consist or detect the analytes like toxins, tumor biomarkers, and viruses. They are commonly used in environmental monitoring because they offer direct and real-time analysis. Real-time means they analyze the particle at the time of analysis, not the delayed analysis. And different phenomena like wave fluorescence are used to detect the interaction of bioelement and the analyte to generate the results. The examples of optical biosensors will be discussed in the upcoming slides. Now, the second type is electrochemical biosensors. As the name suggests, electro and chemical, they consist of the electrodes which are responsible for the positive and negative charges flow or generating the positive and negative charges when the biochemical reaction occur in the process of detection of the analyte. And the type of electric signal that are being generated by using electrochemical biosensors are voltage or current, which is measured in terms of amperes or represented in terms of amperes. A common example of electrochemical biosensors is blood glucose test strip, in which the blood is being recognized by the electrodes inside the sensor, and these signals are received by the transducer, which convert them into the signals that show the readings of blood glucose level inside the sample inserted. The third type is mass-based biosensors, which is further divided into two types, piezoelectric and magnetoelectric types. Piezoelectric biosensors are also known as acoustic biosensors since they are based on acoustic principle sound. And crystals are also used in this type of biosensors, will help certain molecules to absorb on the surface, alter frequencies which help in the analysis of the analyte. This is the basic working principle of the piezoelectric biosensors. And the magnetoelectric biosensors are based on the principle of magnetoelectric effect, which is the coupling between applied magnetic field and generated electric signals in the form of potential difference or electron EV, electron volt. Now come to the classification that is based on biorecognition elements such as antibody, phage or virus, DNA, enzyme and biomimetic compounds. For the detection of biorecognition element such as an antibody, we use immunobiosensors, which consist of unlabeled antibodies that are coupled with the potentiometer. And we also use whole cell biosensors, which consist of the dead microbial cells for the identification of toxins. And the whole cells derived from cyanobacteria are used for the toxin detection. And why they are used? Because they optimize the biodegradation of hazardous substances that are present in the environment. There is also an interesting application of biosensors which is used in the identification of biomimetic material. Now, what are biomimetic material? These are the compounds or material that are man-made or a replica of natural biological objects. For example, honeycomb structure of beehive, strength of spider scales, etc. So in this slide, we are going to discuss about the components that are involved in making biosensors suitable for environmental monitoring, their workflow, and the working principles. As you can see in the diagram mentioned, the first thing is analyte. The analyte is a substance that is going to be analyzed and which is the main element or chief element in the working of biosensors. We must need an analyte, obviously. Analyte can be any toxin or any other contaminant, any compound, harmful compound, right? So what is the next step? The next step is this analyte first react with a biorecognition element, which is another important component in the biosensor. This biorecognition element can be an enzyme, an antibody, a microorganism like bacteria. And the main purpose of this biorecognition element is to provide biochemical reaction with the analyte. This provides the basis for the working of biosensors. Now next come the physicochemical transducer, which is a physical component of a biosensors, and its purpose is to convert the biological response into measurable signals. The biological response is the biochemical reaction that take place by the help of enzyme with reaction with the analyte. The transducer can work on the basis of electroactive substances, pH change, heat, light, fluorescence, mass changes, etc. And then the transducer convert the biochemical reactions that are derived from the biorecognition element into the signals that can be detected by a detector, which is the signal identification component of the biosensors. And after the generation of signals, these signals are transferred to a computer screen so we can see or analyze the results. And the results are obtained or calculated on the basis of the calibration curve. This calibration curve is a way to identify the concentration of an substance. These curves use data points of known substances at varying concentrations, and researchers or developers can use this curve to find the concentration of an analyte in a sample like soil, water, air, etc. So now here is a comparison between biosensor and biomarker, as these two terms are quite similar or can be different from each other. So let's see on what basis these are different from each other. Let's consider an example of a thermometer. Why we use thermometer? We use thermometer to measure the body heat and the amount of body heat generated in a particular body of the living organism. Now in this example, what will be the biomarker? Biomarker provide the indication, like we can say that the heat that is generated inside the body act as an indicator that something is going wrong, and this will be the biomarker. And what will be the biosensor? Biosensor will be the thermometer, which consists of the mercury as the recognition element, which will react with the heat and then generate the results and show the calibration. This calibration is not in the graphical forms but in the form of temperatures that are being mentioned on the thermometer. So I hope with the help of this example you can easily differentiate between the biosensor and biomarker. So here is a list of environmental contaminants that biosensors can detect. This include heavy metals, nitrogenous compounds, phenolic compounds, PAH like polycyclic aromatic hydrocarbons and organophosphorus compounds such as those in pesticides and herbicides. Heavy metals can be classified into useful and harmful types. The useful are zinc and copper, which are required by our body for the better metabolism, and the harmful ones are mercury, lead, etc., which can cause carcinogenic effect on the tissues of the body and they cause cancer. The nitrogenous compound involve nitrites, which are the intermediate products. And why they are considered bad? Because if they are swallowed, they react in the acidic environment and form nitrosamines, which are the carcinogenic compounds. The next is phenolic compounds or phenol toxicity, which is due to the exposure of phenol in the explosive production industry, and this is also a harmful chemical reaction which can cause necrosis, which is the unprogrammed cell death, and the phenolic toxicity is also known as protoplasmic poison. And as we all know, what is protoplasm? It is a mixture of various organic and inorganic substances that make up the living nucleus, cytoplasm, and other organelles of the body of the cell. And another example of environmental contaminants is polycyclic aromatic hydrocarbons, which are the main toxic compounds that are found in marine environments. And an example of such compound is pyrene, which is released when the plastic degradation occur inside the water. And the last one is organophosphorus compounds, which are the harmful chemicals that are found in pesticides and herbicides. So here come the most important and exciting part of my presentation, which is the application of biosensors in environmental monitoring. And here I covered 4 examples, and I'm going to explain this. The number 1 is toxic compounds detection. In this type of detection, we use a microtox test, which is an in vitro testing system that is based on the principle of bioluminescence or fluorescence that is generated by using bioluminescent bacteria such as marine bacterium Vibrio fischeri. And the bacteria which is being used in the identification of toxins is working on the principle of inhibition. And here we also discussed the application of genetic engineering in generating the bioluminescence property in the bacteria. As I mentioned in the example that a Japanese team generated this technology or of the fluorescence by using the firefly luciferase genes, which are responsible for the luminescence in fireflies, and the genetic recombination occur in the E. coli bacteria. This is also an example of biomimetic compounds or biomimicry. Then in the next point, the further process is explained that E. coli cells were immobilized on magnetic beads. Why we are using magnetic beads? Because they are recoverable and enzyme can be immobilized on them very safely. And in the last point, as I discussed earlier, that the toxic compound detection by using the bioluminescence bacteria or microorganism is based on the principle of inhibition. So the toxic compound inhibit the resultant light from the solution containing sample of bacteria and the luciferin. And as the sample is in the liquid form, so we are using a flow through sensor system, which is an electronic device that measure or regulate the flow rate of liquids or gases within the pipes or tubes. And as we see, there are mentioned some readings like light reduction of 12% were achieved by the sample containing 0.1 part per million midazoline, which is a plant growth regulator or a herbicide. And this activity also resulted in the detection of compounds, toxic compounds from the isobutyron, which is a herbicide that is used in wheat crop seed during seed germination. And ioxanil and propanil are also types of herbicides. So this activity can detect very trace amount of toxins, as we mentioned in the uh, concentrations like 50 part per billion and zero point 0.1 part per million. So this activity is quite impactful in detecting very trace amounts of toxins that are present in the herbicides. And now here is another application of fluorescence based biosensors in the detection of heavy metals. We have discussed the few examples of heavy metals in terms of useful and harmful. But what are basically heavy metals? These are the compounds which have relatively high mass to density ratio or density, and due to this property they are non biodegradable, i.e., they cannot be easily degraded into simpler and non toxic compounds. That's why they are the dangerous, or we can say the most dangerous environmental contaminants. And it include mercury, cadmium, lead, chromium, and certain amounts, high amounts of zinc and copper. They can accumulate in the abiotic components of the environment, like in soil, in water, and they can be released in air and can cause carcinogenic effect on the life of living organisms. So for the detection of heavy metals, we can use the enzyme acid phosphatase, which is specific for mercury, chromium, and copper detection. And this type of biosensor is also based on the principle of inhibition, and the fluorescence decrease as it detect the high amounts or high concentrations of the toxins or heavy metals. The enzyme acid phosphatase is specifically used for the detection of heavy metals because of its stability for more than 2 months at a specific temperature of 4 degrees Celsius. It is also observed that this type of biosensors can also detect the mixture of heavy metals that are being present inside an analyte sample. Now, this is another exciting example of biosensors which are being used in the detection of nitrogenous compounds, for example, nitrites, by using the biosensors. Nitrites are intermediate compounds that are found in soil, and they have to be fixed by the bacteria in order to be converted into nitrates, which is then used for the plants as a source of providing the nitrogen. So the biosensor designed for the detection of nitrites is an example of the electrochemical biosensors because it uses a protein which is cytochrome C, and amperometric measurements are involved. So we can say that these elements contribute to the formation of a electrochemical biosensor. The cytochrome C protein is found in mitochondria, and it is involved in oxidative phosphorylation and role in electron transport chain. This cytochrome C protein react with the nitrite and make a compound called CCNIR, cytochrome C nitrite. A linear dependency is observed, which is defined as having at least one linear combination of elements. Like the detection is based on the concentration and the combination of how much the amount of cytochrome C and nitrites is made. And here the concentration of nitrites are mentioned 0.015 and 2.35 micrometer, and the detection limit of this combination is 4 nanometer. The detection limit is defined as the lowest concentration of the analyte, which means this system can also detect the combination having the lowest concentration of the analyte nitride, as well as the combination which consists of the highest amount of the nitride and cytochrome C. And this is the last example of biosensors which are applied to the monitoring of phenolic compounds, and we discussed that phenolic compounds are protoplasmic poison, and the toxicity they contain is due to the exposure in explosive production industries, and also production of paper pulp, plastic, dyes, drugs, oil refinery, polymers, etc. And when they are exposed to the environments, they become organic pollutants, and their concentration, even they are less than 1 part per million, can have disastrous effect on the health of living organisms. Certain enzymes can also be used for the detection of phenolic components such as lactase or tyrosinase, or the DNA of these enzymes are being used. And this is another application of electrochemical biosensors, in which the chemical compound or component is the DNA of the enzymes, and the electric signals are amperometric in nature, which will help in the detection of the phenols. The double-stranded DNA is immobilized on a surface of disposable carbon electrode, which will generate the amperometric signals for the detection of phenolic compounds that are present in the sample, such as water, or it can be a soil sample. Okay, so now let's consider the superiority of environmental biosensors over conventional monitoring methods, which are GIS, Geographic Information System, which is basically used for monitoring the air quality, and the Universal Soil Loss Equation USLE, which is used to predict the soil loss due to water erosion, and many other systems. As I mentioned, that biosensors are used for environmental monitoring have several advantages over the conventional systems, and some of these advantages are their ability for portability, means we can carry a biosensor with ourselves at any place and at any time, and these biosensor will provide the real time analysis of the analyte, which is the main advantage of using environmental biosensors over conventional monitoring systems. This ability of portability is come from the miniaturization of these biosensors, which means to make a biosensor in such a small size that it can be easily carried into the hand of a person. The next point is measurement of a pollutant with minimal samples, which means we do not need a large amount of sample for the working of biosensor. Instead, we use a specific quantity of a sample, and we can easily analyze them by using biosensors, environmental biosensors. And the final point is high specificity and sensitivity, which means that environmental biosensors can detect very specific analyte present in the sample, as we have discussed earlier in the examples, that heavy metals, phenolic compounds, nitrogenous compounds can be separately identified by different kind of biosensors, and they are very sensitive, which means they can detect very small or part per million or part per billion quantity of the analyte that is present in the sample. So all these factors increase the importance of biosensors. So here is the summary of the lecture, as we have discussed in detail about the introduction of biosensors, their working, components involved, examples specifically related to our topic, and I mentioned few concerns in the summary related to the biosensors, which is linked with the modern environmental monitoring needs. As the environment is polluting itself at a very high rate, so we need a rapid analysis system which is provided by the biosensors. And to design a biosensors, one must have good knowledge of biology, chemistry, kinetics, electrochemistry, physics. And by applying all these knowledge in a proper way, a good biosensor can be designed. These are the references which I have used for making this lecture. Thank you so much for listening, and all your queries and questions are welcome in the comment section. Thank you so much.